Our next presentation will be by Daniel Geschwind, a Gordon and Virginia McDonald Distinguished Professor in Neurology, Psychiatry, and Human Genetics at UCLA.
Let's see if I can use this. Anyway, thanks so much again for inviting me to the symposium. It's great to have been have the opportunity to also be working on this project. Let me get that. Is that? So, before I get started, the work that I'm going to talk about is using transcriptomics and epigenetics to try to accelerate aging in a dish. And kind of what's been pushing us in this direction, and this is a collaboration with Steve Horvath, so I'll be presenting work that we've been doing together and some work that he's been doing as well. And as you probably know, methods using especially mouse models, model systems haven't been so successful in translating disorders of aging and dementia, neurodegenerative diseases into the clinic. And so people, the huge advance in vitro systems and stem cell biology to begin to create tissues in a dish. And I'll present to you some of the work that kind of started us thinking in this direction. What are some of the issues with the tissues in the dish, and how do we age them? So this is a work in progress. It's not a completed story. And as I'll tell you, some of the thinking that we started this with was, in retrospect, very naive. And but we took a very simplistic approach, and I'll share that with you. Whoops. So this is the Mac to PC transition, but I'll, basically the first, when we started to use these models, especially to even study autism in a dish, one of the questions that hadn't been answered at that point was how well do they, at a really comprehensive level, do they match in vivo development? And there were tools like BrainSpan that had come along where we were peripherally involved, but it was primarily driven by Nanad Sestan and folks at the Allen Institute, including Ed Lean, who profiled human cortical development over time from 8 weeks gestation through adulthood. And that would, when we reason, that gives you an in vivo gold standard that you're trying to match in the dish. And so we developed, we just asked a very simple question. Could we develop methods using in vivo transcriptomes that would inform the development of in vitro transcriptomes? In other words, how well do my cells model in vitro development as what is modeled? And this is showing the BrainSpan stages on the left, where birth is about 8 and 13 is 20 years of age. So after birth is kind of 9 and 10, it's childhood and adolescence, et cetera. And mid-fetal gestation is stage 5. So what you can't really see here, but what you'll have to believe me about is, so we developed 3 basic tools. One was a very simple rank-rank hypergeometric test, which just asked, if you have transitions in vitro, what do they match in vivo? And so what transitions are best matched? And so that's shown there where, essentially if you have red in both the right upper quadrant and left lower quadrant, that would be very, very strong matching. We developed a machine learning tool based on multiple data sets using, first discovery and then test and replication data sets, where we could fairly accurately determine the developmental maturity and regional identity of the tissue based on this BrainSpan in vivo data. And then, and this is just showing it's a little hard to see, but the performance and how many data sets we use to test and develop this simple black box machine learning tool. The third thing we did is to ask what biological processes are conserved. And so we used, actually, this is another tool. This is in Steve's previous career. The first 10 years of his career, he developed, and we collaborated with him in some of this really beautiful work where he developed this tool called whole genome co-expression network analysis. And the idea here is that you use graph theoretical measures to identify modules of co-expressed genes that correspond to basic biological processes. And what we could see that when we were looking at two-dimensional in vitro neural progenitor cultures that we could model basic developmental processes like neuronal migration. This is showing the kind of what happens over time during in vivo development. And so you have progenitors and cell cycle going down, nervous system development, nervous system processes going up, various aspects of synaptic transmission going slowly up over time, first starting with adhesion molecules and then moving outward. And so we could see that these were conserved. And interestingly to us, this midnight blue module contained a lot of chromatin histone modifiers. And we were very interested in this because when we looked at the autism candidate genes that had been identified by sequencing at that point, they were highly conserved and present in that module, which told us, well, we can model early development. But one of the things we learned from this work was that using cell lines and actually iPS-derived neurons, primary progenitors from fetal brain, that most of the cultures didn't get past somewhere between 20 and 25 weeks in vitro, even if they were grown for 8, 12, 16 weeks. And we got cells from Ricardo Dolmetsch's lab, Rusty Gage's lab, et cetera, and we tested this. And this is kind of showing that this is what these maps are supposed to look like. And you can basically see the primary neural progenitors were actually not doing badly. They're getting, what we're doing here is comparing stage 1 to 2, 1 to 3, 1 to 4. In other words, this is the earliest to latest, and the best transition is kind of 1 to 6 here. When we looked at iPSC-derived cultures, they were even a little bit more immature, and this is from Allen brain data from in vivo cortical laminae, a Miller et al. Paper in Nature several years ago. And so this kind of started us thinking, how do we make these more mature? And we took a really simplistic approach, which is we can basically take the modules that are conserved, identify bioinformatically the transcription factors that are putatively driving them, and then ask what's present in vivo that's absent in vitro, kind of a subtraction. And then we can overexpress those or knock those out that we need to. And so we started on that, and this is just an example of several of them. So focus on identifying the signatures, identify candidate drivers, and then see if we can do this. So this is just showing a cortical transcriptome of one transcription factor and another one, and what the progenitors look like over time. And so these would be, now of course, there are many things happening here. So we just heard about developmental age, we heard about epigenetic age, and what's happening here also in vivo is the cell type composition is changing massively. And so what we're missing in a lot of these cultures are certain of the different neuronal types as well as glial types. So we might expect that these may not increase aging necessarily, but kind of increase how this matches the stage. And indeed, that's kind of what we're beginning to see, that this overly simplistic approach that we hoped would work, the jury's out, but so far, I'll give you an example on, in a second. As we were doing this, Sergio Pasca and other laboratories developed 3D cultures, and this is a very simple 3D culture called human forebrain spheroids. And I'm showing, again, an overlap here. This is work that we did with Sergio showing that these get slightly more mature after just about 10 weeks. They're already looking like 12 to 16 week 2D cultures. And so what we reasoned is, and Sergio's lab and others have shown that these, interestingly map on to structures very grossly, not as nicely as the retina in vitro, but grossly one can begin to see laminated or at least pattern structures in vivo. And so we began to realize that this culture system would be better than using the two-dimensional cultures, so we kind of switched to this. And essentially what we've been doing is overexpressing. This is the BCL6 and SAPB1 just as examples, sorting out the early cells at a very, very early stage, and then growing the organoids or the forebrain spheroids from them. One of the points I want to make is that we use a very directed differentiation to create a dorsal telencephalon. We're creating a specific kind of structure, and this increases the reliability of these cultures enormously. And this is all based on Sergio's work. And so what we see, interestingly, and again, let's say this is SATB1 overexpression, what we see is a decrease in the green here in the forebrain kind of dorsal telencephalon markers and an increase in interneuron ventral forebrain markers. So what we've essentially done is, and I'll show this in a couple, this is actually a immuno showing us, normally we don't get any somatostatin or parvalbumin interneurons at this stage, and here what's happened is we've driven these neurons that are, in a forebrain system. What's interesting is that SATB1 had been shown to be important for interneuron maturation by Gord Fischel's lab, but this actually shows that just even in the context of factors that are dorsalizing, we're actually driving the whole culture to a dorsal identity, that the overexpression of this single transcription factor is able to overcome that, and drive these neurons to inhibitory. But it tells us that essentially a lot of the transcription, using transcription factors, a lot of what we're, unless we get lucky, a lot of what we're going to be doing is driving cell fates in vitro. In other words, changing the composition rather than changing the maturation. So that's essentially here. So we're increasing ventral progenitors. And so with some of the other things that we've overexpressed, we've begun to see similar things where we're actually driving gliogenesis early rather than late. So again, this kind of simplistic idea that maybe we would be able to just identify the several dozen transcription factors that are missing. Yes, we identified them. Yes, they do interesting biology, but they're not increasing the maturation per se of the cells that are there. They're actually increasing the maturation of the tissue by increasing the complexity of the cell types in it. So you just, so you just heard Tom gave a really great introduction to this notion of an epigenetic clock that Steve Horvath first observed in 2013. And so what I'm showing here is much longer differentiations from 0 now out to 600 days done in Sergio Pasca's lab and some in our lab, where we look at DNA methylation age, and the colors are different individuals. So we can see that DNA methylation age is highly correlated with differentiation day, and predicted age of birth would be somewhere around 281 days, based on this. So it's within the 95% confidence interval, encompasses actual, birth based on, this is from humans. So it's pretty interesting, and again, as Tom showed and as Steve has showed before, that we know in vivo this clock is preserved during development and fetal development, and we can use it here to show that these cultures, as they're on human time, they're on their own developmental clock, but they are developing, and we are, after about 250, 260 days, close to 300 days, getting cultures that are mature, compared to previous cultures. This is showing a rank-rank hypergeometric based on gene expression, and this is showing prenatal and postnatal. And we'd never seen anything like out, past 6 before, really even past 5. And what we're seeing is that as these cultures, develop past 300 days, they really become looking like postnatal brain based on this, on the in vivo data. So that's transcriptomic data showing that they're developing kind of as would predicted based on their internal clock in vitro. So the good news is we can grow more mature cultures. The bad news, especially for postdocs, is that it takes 270 days. On the other hand, that may be faster than some mouse experiments. But of course, we still really need to accelerate this, and I think it's possible to accelerate this. This is just showing some of the module trajectories that show that what we find really does also follow in vivo trajectories. In other words, the processes that are going on, like cell cycle, synaptogenesis, gliogenesis, et cetera, follow what's happening in vivo. And so we have a nice system. This is just a kind of getting more into neurophysiology. We're actually patching on these cultures now, Sergio is. And when we can show that there's normally a switch in the NMDA subunits and AMPA subunits around birth, and we're actually seeing that, and we're seeing many of the other maturation features. So this is good. It depends whether you're glass half full or half empty kind of person, but it shows that if you grow these long enough and you grow them in the appropriate conditions, we can actually get relatively mature cultures. But how about the, really aging these, like getting older cultures? Can we kind of push the methylation age or accelerate it? And so what Steve had done, and we had collaborated with him a little bit on this and helped out, he had done work showing that acceleration or deceleration, your delta in methylation age or relative to chronologic age is heritable. And so he then reasoned he can do a genome-wide association study on that quantitative phenotype to identify loci that would cause, that should be causal with regards to age acceleration or deceleration, and that was done. And there are a bunch of genes that were implicated, especially this gene MLST8 and DHX57. And so we reasoned that we'll overexpress these in HNP cells. Now we've gone to the 3D organoids. And I have a compulsive postdoctoral fellow who is saving everything for last. So we've been doing everything in a randomized way, and we're making all the libraries at once and collecting it. But in the next actual 3 months, this will be completed. What's interesting is that we were originally only going to go to about 120 days, but because of what we saw in vitro with Sergio's, we decided to go to 200 to push, to get more power. So this is ongoing, and hopefully, if there is a next time where, I'll actually be able to present, the entire, what we've actually found. But I also wanted to tell you 2 updates on some really interesting work that Steve has been doing, and he may talk about this a little bit later as well. And is that as he begins to refine this, he's been looking at the epigenetic clock and did a GWAS in blood, and this is in I think about 9,000 subjects or so. It's a huge GWAS, really well powered. And so what he's shown before is that this methylation clock is the most accurate predictor of chronologic age, much more accurate than, let's say, telomere length. But we know that telomere length also predicts, but with about half the accuracy. And what was interesting is he found a locus here that overlapped with TERT, and this is looking at the intrinsic epigenetic age acceleration. And this is an overlay plot showing the p-value for intrinsic epigenetic age acceleration and for lengthening telomeres. Totally overlapping. In fact, I think 3 of these SNPs are shared. And, but what's really remarkable about it is that it's kind of going in a funny direction, that genetic variants in TERT that are associated with longer telomeres were associated with higher intrinsic epigenetic age acceleration. So the genetic variants in TERT that associated with younger biological age, longer telomeres, are associated with older biological age according to the epigenetic clock. And again, this just gets to the fact that this issue of kind of aging is going to turn out to be a very, very, as Tom just showed you, there's kind of maturation and there's aging, but even within aging, Steve and other people have shown there are extrinsic clocks, intrinsic clocks, and I think these kind of factors are going to show some really interesting complexity. So Steve's group, he's working with others to try to dissect this, and as genes are found, we're testing them, we plan to test them in vitro in these stem cell models to see if we can accelerate age in vitro in these models and hopefully have better models, improve the models of in vitro aging. So this is Sergio Pasca and Steve Horvath. I mentioned the BrainSpan Consortium, and again, this is a kind of shout out to kind of shared large-scale data resources, because without this, it would be very difficult to actually have an in vivo comparator. The folks in my lab, Jason Stein and Luis de la Torre Bieda, did the original context work and machine learning. Aaron has been driving the recent 3D projects along with Zainab in the lab. And then in our original study, these were the folks who provided us with their either their cells or their gene expression data so that we could actually ask how old they were. Anyway, thanks for your attention.
We have time for 1 question. Microphone's coming to you.
I was almost on time.
It seems in a lot of these studies you focus on cell autonomous genes expressed rather than things that might be secreted or affect the matrix or these sorts of things. And I wonder if you put your attention on some of these other features, whether that will focus on the other half of the problem probably.
Sure. No, I think it's a great question. We're not overlooking that. Our logic is kind of this, just like with that SAT, the SAT experiment. All the extrinsic factors are driving it to dorsal fate, and yet, overexpression of this one intrinsic factor leads it to a ventral fate. So that, and just like the Yamanaka experiments, we reason that, of course, external environment is absolutely critical, but that you can drive things using intrinsic transcriptional programs. That's our kind of hypothesis. It's not ignoring that other piece, but asking whether we can change the state to make it from kind of younger to older, to accelerate the aging, using intrinsic factors. But that doesn't mean it wouldn't be interesting to look at the extrinsic factors. It's just that if we have modules that correspond to, let's say, things that we know are critical for neuronal maturation, like synaptogenesis, that include all of those isoform changes that are critical for maturation, we can identify the microRNA or transcription factors that are driving those. And so it gives us a kind of, a handle from the kind of bioinformatics into, driving things intrinsically. But I acknowledge it's just a start. One has to start somewhere. And so we've started, with that kind of logic. But it doesn't mean that focusing entirely on the extrinsic factors also wouldn't be useful.
Thank you so much.